Okay, so we're going to start with the very first cell, which is the product of the abiotic origin of cellular life. And this is going to rapidly split into the archaea and the bacteria. This basically happens right there at the very first juncture as we imagine it. The archaea subsequently split up into several groups, including some more basal groups. The core archaeota is recognized as one of these. And the two main clades of archaea are the eukaryote and the prokaryote. Within the eukaryotes, the two groups that we identified by name or by, I showed you a picture of these, were the methanogens and the extreme halophiles. Whereas within the prokaryote, we talked pretty much about these really bizarre organisms living in hot acidic conditions. And we also suppose that the original eukaryotic cell, or the eocyte, the Dawn cell, might have come from this lineage as well. One of the points that I emphasize is that we tend to focus on these kind of exotic types, methanogens, extreme halophiles, and thermocytophiles, but the real truth is that the majority of the archaea are not so exotic. They don't live under extreme conditions. They're just like regular old cells. It just happens that the ones that catch our attention are the ones that are really kind of bizarre. And now as we move down to the domain bacteria, we have a couple of basal groups, the spirochetes and the chlamydiae, and then we've got the cyanobacteria, the firmicutes, and the proteobacteria. Spirochetes, the ones we typically name here, the Treponema pallidum, the cause of syphilis, and Borrelia burgdorferi, which is the agent associated with Lyme disease. The chlamydiae are those really tiny obligate intracellular parasites, and in this group we identified, I think, Chlamydia trachomatis. Sometimes I'll do Chlamydia pneumoniae or Chlamydia psittaci. There are several species here. Within the cyanobacteria, usually I'll identify spirulina. Within the firmicutes, we've got a couple of organisms that we recognize from laboratory, the Staphylococcus and the Streptococcus. And then within the proteobacteria, we've got alphas, the betas, the gammas, the deltas, and the epsilons, of which we have a couple from laboratory that we recognize, E. coli from the gamma bacteria, the gamma proteobacteria, as well as Pseudomonas, also from the gamma proteobacteria. And that pretty much takes care of our picture of the prokaryotic domains. And as we pointed out in class, the diversification of the prokaryotic domains is taking place pretty much entirely within the Archean, the early Archean, middle Archean, and late Archean. And then we have the great oxygenation event taking place around 2.5 billion years ago, which is the start of the Proterozoic. And it's really around that time where we see the origin of the very first eukaryotic cell. Eukaryotic domain is presumably coming about around the earliest parts of the Proterozoic. And given the story of endosymbiosis, as well as the Deduve picture on the origin of nucleated cells, we sort of picture this guy as being mitochondriate, meaning that as far as we know, there are no primitively amitochondriate eukaryotes. And then we talked about three clades, the Archoplacida, the Excavata, and the SAR group, which includes the Straminopiles, the Alveolata, and the Rhizaria. We sometimes cluster these together and call them the biconts, because they have two flagella. And the archoplastids are characterized by this is a lineage that gives rise to plants. And so we talk about in laboratory, we'll be developing this a little bit later. For now, we'll identify a few of the protistan members of the archoplastids, the red algae or the rhodophyta. Then we can talk about the green plants, and the green algae are half of the green plants, and the other half are the streptophytes. And then within the streptophytes, we've got some basal, still protistan streptophytes, as well as the plants. And in the excavata, we've got the diplomonads and the parabasalids on one lineage. Those are the ones that have lost their mitochondria secondarily. And then on the other side, we've got the euglenozoa, which include things like euglena, an algal-like protist, is photosynthetic, lives in pond water, as well as the cnidoplastids, including trypanosomes, these blood and tissue parasites that you find in places like Africa and South America. And so in the third clade, the SAR group, we've got a basal lineage, the Rhizaria, which include things like the radiolarians and the foraminifera, which we discussed before. And then we've got the two crown groups in the SAR clade, the Straminopiles, which are represented by things like diatoms, and then you've got the brown algae like the macrocystis kelps that we have off of the California coast, and then the oomycetes, the oomycota, which is most notably represented by Phytophthora, which is a plant parasite and the agent that caused the Irish potato famine. And the last group of bicons are the alveolata, which include things like the dinoflagellates, ciliates, and apicomplexa. The dinoflagellates are the things that include the Lingulodinium, the red tide organism that we see periodically off the California coast. And then within the apicomplexa, we've got Toxoplasma, the agent of toxoplasmosis, as well as Plasmodium, which is the agent of malaria. And in the other main lineage of eukaryotes, the ones that have single flagella, are the uniconta, in which we've got two larger clades, the amoebozoa, largely unicellular things, but also the slime molds are there, and the opisthoconta, which are characterized by having flagella at the rear of the cell. Within the amoebozoa, we talked about single-celled amoebae like Entamoeba histolytica and the acanthamoeba, the one that causes the eye-eating disease. And then we discussed two different kinds of slime molds. There's one called the plasmodial slime mold, no relation to Plasmodium, the apicomplexan, but it's called the plasmodial slime mold, which doesn't have any true cellular structure. And then we've got the dictyostelium, are called cellular slime molds. Those are the ones that have the amoeba-like units that come together to form a little slug, which then metamorphose into something more spectacular. And then on the other lineage of the uniconts, we've got the opisthoconta, and they're going to be split into the animals. We'll talk about them later. And then the coanflagellates, which are closely related to animals, but they're still protists. And then we've got the fungi. And that pretty much covers the layout of the basal parts of the tree of life. Now within the fungi, we're going to zoom in on them now. We identified 5 clades: the Chytridiomycota, the Zygomycota, the Glomeromycota, and then the crown groups, the Ascomycota and the Basidiomycota. We'll draw them in like this. And some of the names we associated with the different groups of fungi. With the chytrids, we identified the frog disease. With the Zygomycetes, we identified the organisms that causes white bread mold, as well as the organism that makes us tempeh. Within the Glomeromycota, we were talking at length about endomycorrhizae in laboratory, and those are all glomus species. And then within the Ascomycota, there are several examples. Truffles and Saccharomyces are a couple that we named, and then we've got the mushrooms and organisms like Huitlacoche in the Basidiomycota. Alright, so let's take a look at this tree. And there you have it. And so what we'll be doing next is putting some detail onto plants. I would do it here, except I don't really have enough space on this diagram.